a nice little friendly G protein coupled receptor. In reality, remember, they're crazy proteins and they're twisted around and they're elegant in their twistings and they're really quite organized and beautiful. If you ever look at the actual structural pictures of them, which actually I encourage you guys to do. Visit your old Uncle Google later and see some of the protein structures are. It's really quite nice. But the point is that these are proteins, and they're expressed in, remember we talked about the central dogma DNA, RNA production into protein. So you're going to have different DNA in different olfactory receptive cells that is going to produce different proteins that get inserted in the membrane and become these channels, or excuse me, not channels, receptors. And so you can have cells that we can tag for different genes, and they mean they have different receptors. Those different receptors are going to be sensitive to flowers or barbecue or coffee or whatever your smells are. Okay? And so that's kind of what we're looking at here. That's all this color map is just distinguishing. Different categories of genes can be spatially located in sort of an organized format. And what we can see here is a nicer way to look at that. So here we have that olfactory epithelium. Okay, so remember, above here, they're going to project and go through the cribriform plate into the olfactory bulb, right? Should I slow down here, take a breath, make sure everyone's on the same page? Everybody knows what we're looking at? Okay. Sorry, I just get so excited about these stuff sometimes.
You're talking again?
No. Gee, thank you very much. It's, I get all the kick I need from neuroscience. I almost quit Sinatra and said I get no kick from champagne. Yes.
That would have been a good one.
Huh?
That would have been a good one.
Yeah. Mere alcohol doesn't do it all. You guys know what I'm talking about. It's something called I get a kick out of you. Anyways, so here's our olfactory epithelium. And the colors here are showing different cells that have different genes coding for different receptors that detect different odors. It's all three different ways of saying the same thing, right? If I'm talking about different genes, or if I'm talking about different receptors, I'm talking about different odors. It's all, I'm saying the same thing, I'm just saying it 3 ways so that I can bring everyone together who likes it in different ways. And so, just like we started off class, right? We looked at a very similar picture of this for the taste cell. We drew a very similar picture of this with our cone cells, right? And what we can see is that we have different smells now. Citrus, floral, peppermint, almond. Okay? And we are measuring, right, the electrical potentials of these different cells. Notice that the colors correspond nicely to the cell. So this must be a green cell. These blue trains are this blue cell, and these red trains are this red cell. Okay? So all we're doing is we're doing this, but now we're doing it times 3. And we're doing it not just times 3, but with 3 different odors for 3 different types of cells. Now if they just made 3 different yellow cells, then you'd have to maintain in your mind the memory that these 3 different yellow cells have 3 different genes and proteins in them, right? But they save you that trouble, and they color code it. So that's why it looks this way.
Are those action potentials?
Yeah, I would imagine that they must be based on the shape. In fact, it looks like it's probably both, right? It looks like we have a graded potential and with a superimposed action potential. So it's showing the graded potential that you're recording in the receptors, the cilia, with the action potentials generated by that, superimposed on that, based on probably a second recording up here. You see how that makes sense? Yeah? Okay. And it's really helpful because if you want to walk it backwards, it'll show you where the threshold is, right? It's going to be just wherever that is, wherever that spot is. So anyways, the point is we have different patterns, right? So, but they're all responding to different odors, just in different ways, right? And so it's not just that, and I used to say this about the chemoreception lecture, but it's that all cells like pizza. Some cells have different preferences for flavor. Some cells like sausage, some like pepperoni, some like cheese, some like anchovies, right? Everybody likes ice cream. Some cells have their favorite flavor. So same thing here. And just like we talked about, how do we detect which of these is in the environment that we're smelling? Well, it's by the combinations of these patterns. So you're never going to know which of these cells or smells is, smell is a perceptual thing. You're never going to know which odor is in the environment that you're smelling. If all you do as the brain is look at these cells' input, because they fire for all of them. You're never really going to be able to distinguish, right? Yeah.
What about the almond one for the green?
What about it?
I mean, does that just mean that that receptor isn't sensitive to the smell of almond or to the odor of almond?
It does mean that.
So you said they all react to it, so it still reacts, it's just like not enough to picture, or?
You're right. I am wrong. That one does not react.
Oh. Yeah, 'cause I was like, is it just like
Yeah.
super small, or?
I mean, I was just kind of speaking generically, but you're right. Absolutely. Yeah, this one just. But so even so, if say this one doesn't respond, but if you're the brain and all you're getting is this,
Mhm.
how do you know? Right? And so that's the way you're going to know is by this combined with the patterns that you're receiving from these other groups. Right?
It makes sense now about what you're saying about how they have different preferences.
Yeah.
And why they only.
Absolutely. So this is kind of the idea there. We'll see how this pathway works. So we've moved from having Ron Burgundy's smelly thing, I forget whatever he was smelling, a dirty diaper or something. In the scene.
Wasn't it the cologne?
It was the cologne, it was quite pungent. That's right. So it comes in, cinches the nostrils, it integrates, it's detected by these cilia, right? You have that cascade of signaling from the G protein, GULF, GULF, right? So there's that downstream signaling cascade with cyclase, ATP, cyclic AMP, right? Opening the sodium, the calcium channels, leading to efflux of chloride channels. And now we've looked at the action potentials that are generated after degraded potentials are generated. Now we're moving beyond these cells, which is what we've been at here. And we're going to see what happens next. So now we're crossing through the cribriform plate. These axons project, and it's similar. It's similar to the model that we kind of had in mind when we looked at the retinal cells, or excuse me, the visual system, when we saw this network from photoreceptor to bipolar to retinal ganglion cells, and it also has lateral inhibition like we saw with endocrine horizontal cells. Okay, so you're going to get these receptor cells. They're going to make a projection, okay, onto these other types of olfactory cells. We're going to call them secondary olfactory neurons. Okay, now they're going to make projections to these, and every one of these cells receives input from lots of different olfactory receptor neurons. So it's a convergent zone. But the thing is where these converge, all these groupings of synapses, they're called glomeruli. A single one of them is called a glomerulus. Okay? And so each of these glomeruli integrating the connections of lots of different olfactory receptor neurons into a single convergence point onto a cell. The thing about glomeruli is each glomeruli has inputs from the same types of odor receptor neurons. By type, I mean genetically, the type of receptor that it has to detect a particular type of odor.
So probably like that first group would be like the blue, and then the second group of the
Yeah.
yeah, so that would just be blue, and then
blue, red, green, and of course, green representing the different genes.
Yeah.
Yeah. So yeah, and so what you'll see is we have lots of them. Okay, and let's see if we can see some more examples. So just like you were talking about, red, blue, green, right? They're gonna send their projections up to here, but every time, all the convergence zone connections, they're all gonna be from the same type of cell, and they're gonna come from different spots, right? And so you're gonna get glomeruli all over this olfactory bulb, which are these sort of coalescings of the different inputs. And when we have certain odors, right, that are going to activate certain types of receptors, that means it's going to activate certain type of glomeruli. So different patterns. So when we looked at patterns like this of individual cells, you'll have different patterns as well of glomeruli that quote unquote light up given a certain smell. Because any smell is generally a combination of a ton of different smells put together, right? So you're going to see different glomeruli light up. And the different patterns of these glomeruli that, so to speak, light up or get activated is basically going to correspond to the signal of what that smell is that helps us make sense of it in our cortical processing. It's kind of like an example that somebody told me once that has always helped me to make sense of it was that it's like a marquee board. You know what a marquee is? Like a lighting board at like a theater production. You know, it says, yeah, all the light bulbs. It says like, I don't know, movie show tonight, and it says the movie, and it says the thing, and it has like little lights. Maybe it rotates around, and it's kind of from a few generations ago, I guess. But does anybody not know what a marquee lighting sort of board is? Okay, so I'm assuming everyone knows that analogy. And so it'll help you to make sense of it in sort of coding. And so when that happens, this is kind of the projection that I think Corey was trying to draw, right? Okay. And so we start off with our olfactory receptor neuron. It projects through the olfactory receptor or olfactory tract. And just like most of our information in sensory worlds, it will project to the thalamus. However, this is the exception to the rule. The olfactory system is special. It's like a snowflake. Okay? And it will project to the thalamus, but it does not project to the thalamus first. Usually that's our first relay station, isn't it? Instead, it's going to go to a couple other places. Notably piriform cortex, the amygdala, pararhinal cortex, olfactory cortex. And then it will go to the thalamus, particularly the medial dorsal nucleus of the thalamus. So hopefully you can add that to your existing charts. And the thalamus will then, integrate it and do further cortical processing, but it's not its first destination. Usually in sensory systems, it is their first destination. Now from the thalamus, that projection will then go back to cortical areas, particularly this orbital frontal cortex, which is this area, ventral and anterior in the brain. Make sense? But a lot of these direct projections, they're first going to those medial temporal lobe areas. Piriform cortex, perirhinal cortex, amygdala. Amygdala, doesn't that make a lot of sense? Of how strong of an emotional reaction you can have to a signal almost instantaneously before you even make sense of what it is. You're like, oh my God, that's awful. Or you're like, oh my God, I want some. Like, right? Imagine you go into this, like, I don't know, candy factory or, I don't know, whatever smells good. Like, you don't even know what kind of candy it is. You're just like, yeah, just let me follow that trail. You're like a little cartoon character following the whiff of smell, right? And you're like, wherever it is, I just want to go. And you're still kind of figuring out what is it. You just know it smells good, right? And so that connection with the amygdala is going to involve and support a lot of those emotional reactions. Yeah.
Is that smell? So would smell be the emotional reaction to what you have to it, or is like your, because the odor is good, then it smells good?
That's a deep question.
Mhm.
I'm not sure. So smell is not your reaction to it. Your
No.
reaction is your reaction. Your
Right.
emotion is your emotion. Neither of those are the smell. Somehow they're integrating the representation of what that smell is. A lot of what it is going to be, again, more orderly process. That's a little bit higher up, sort of the evolutionary chain. And yeah, and again, sort of tried to be dissociated in that example I gave, where you're just following it because you like the emotion before you've yet figured out what it is, right? By the same token, you can smell something absolutely repulsive, and you just know you need to get the heck away, right? Before you really sort out, like, well, what was it exactly? Was that a sewage system, or was that a cow pasture, or you know, that might take a few moments to figure out. You just know it ain't for you. So, anyways, those are some of our steps. We talked about population coding already, and these maps that can happen, and the sense of, so what you can do is if you can label these different areas, in this case, this is the olfactory bulb of a rodent, okay? You can see these different chemicals, okay, that are really sort of crazy long chemical words, but really represent smells of minty versus fruity, okay? And you can image the different patterns of activation in these core layers, and you can see that they're going to be imaging very different areas that show responsiveness differences. So again, we're seeing this systematic representation of the outside world in our cortical areas. We saw this with tonotopic representation in the auditory cortex, right? We call it tonotopy. We saw it in even the homunculus. Remember we looked at the homunculus for motor systems briefly? And the same thing, we have a somatosensory homunculus. We'll get into that a little bit next week. And we talked about retinotopy maps, right? The systematic representation of the outside world onto our neural tissue, right? Which means it's not just a hodgepodge of, whatever, just goes places and no, there's a systematic organization to it, and so there is sort of a method to the madness there. We talked about the way that information can be conveyed is by temporal patterns of spiking, right? Frequency coding, and we also talked about population coding, right? So again, it's not just frequency, how often it happens, but it's also where the populations are firing that's happening at too. So you have two different kinds of ways that the brain is able to figure out what this information is. Not just how fast it's happening, but also from where is it happening. Okay? That allows a lot of discriminability, which means the ability for the brain to discriminate and figure out what it is. What is that smell of that guy dancing up behind me? Right? Like, is that the smell I want to smell? Or is that maybe a smell like maybe I should get away from? Right? We've got to figure that out. And population coding and frequency coding are two ways that happens. And so these, that's kind of what we're talking about here. Temporal patterns, also evidence in spatial maps. Let me see what we have here. Let me show you, I want to show you a couple other pictures if we have time. Yeah. That is, would you guys like to see other representations of the olfactory system? Okay. So, let's see here. Taste. Motivation. There's Doggy. We'll just kind of do this again. I'm going to move quicker this time because we've covered a lot of introduction elements, and I'm just going to move through this so that we can find some different illustrations that can emphasize the similar points. If it helps to dial it home, okay? So we talked about dogs being great, much greater than we are for their olfactory senses. I was just joking with Erica the other day, saying that I want to send her some old dirty socks of mine so she can give them to our little greyhound puppy Juno, who presumably I still think misses me, and I hope she misses me. And so I want to give her some smells that she likes, right? Because dogs like smells. And it wasn't a crazy idea because Erica's a vet and she didn't tell me it was crazy. She said like, she would have told me that would have not worked. But I haven't done it yet. I'm not really gonna send her myself. Anyways, dogs have good senses of smell. We talked about these introductory elements again. So here's some more images, the same things, right? So you have your nasal cavity here. Here's your cribriform plate. We zoom in on this, and we see our olfactory receptor neurons. They're crossing in and projecting into the olfactory bulb that then gets sent out to the olfactory tract, right? One of those cranial nerves. They're going to be activated through the olfactory epithelium by these airborne molecules. And again, these are our projection areas. Okay, so they're going to come in. This is the olfactory bulb. So we're looking at a ventral view of the brain. We're looking at it from the bottom up. This is that olfactory bulb that Corey drew for us, and our olfactory tract coming in, the cranial nerve. Okay, and its projection areas are not the thalamus primarily. It's these areas: the entorhinal cortex, the amygdala, piriform cortex, something called the olfactory tubercle. Okay. So that's kind of a graphical representation of the anatomy of where some of these areas are that's going to do its direct projections. Again, very medial temporal lobe, right? So this is our frontal lobe here. This is your temporal lobe. This is the medial side of the temporal lobe. Again, an area that we haven't talked a lot about in class yet, about its importance for memory systems, but we'll talk about that in the later sections of the course. And hopefully, we've already dialed in the idea and connecting those dots of how important these projections to the memory systems are for smell, right? And so, you know, can you guys all think of a couple examples of some smells that if you were to smell them right now would take you instantly back to something in your childhood?
Mhm. Yeah.
Right?
Mine would be like cooked bacon and coffee.
Oh yeah. Cooked bacon and coffee. Now you're talking like
Super crispy, like really, really, really cooked.
yeah. Man, don't tempt me. It's delicious. Yeah.
I don't know if this happens, it's a common like phenomenon or something, but sometimes when you're away from home for a real long time, when you come home and you kind of you smell your home, it kind of like
Mom's cooking.
yeah, I don't know, it's weird that you just smell your own house, kind of just,
Yeah.
That's like comforting.
Yeah. That's good. That must mean you don't have too dirty of a house because it doesn't send you running when you come home and smell it. Yeah, Courtney?
The smell of like firewood in a chimney
yeah.
smells like Christmas.
Yeah. That's pretty awesome, isn't it?
Yeah.
Some pine needles or something. Yeah, that's really good. Yeah, I mean, it's almost instantaneous, right? As soon as you smell that, you're like, yep, I got that, right? Direct projection to our medial temporal lobe memory areas. Really important evolutionarily too, right? You're gonna need that type of information. Yeah, I remember when I was a kid in the 80s, my grandpa and my grandma, they used to smoke a lot of cigarettes. She had her Palmolas and he had his Marlboros. And I loved hanging out with Grandpa. He was awesome. He'd always take us to Walgreens and buy us little toys and fun stuff. I actually lost my brother once there. He was mortified. He thought my mom was going to kill him, and she probably would have. And he came in from out of town and took us to go buy a bunch of toys. And he probably got busy buying lottery tickets and lost us running around with our dads. And so I used to get really excited because I never knew what it was, but there was like you said, you go home and it's the smell of home, but it was like when I would ride in his car, he would drive these Cadillac cars around in the 80s, and you know, it's just full of what I now know is just like Marlboro smoke. So you know that smell of like smoking. But for me, it was never a bad smell because it was just the smell of Grandpa. So even now, I smell. I'm like, oh, that's pretty good. It's like being back with Grandpa, right? So again, talking about that association with memory, right? It doesn't give, you know, the emotional valence we give to it, whether it's a gagging smell of smoking or a positive smell of Grandpa, right? That's an emotional valence that the amygdala is giving, right? To an otherwise, a smell is just a smell, it's just neutral, right? So it's going into the entorhinal cortex, which we'll see later on is the pipeline anatomically into the hippocampus for memory formation. So it's going to the entorhinal cortex, but at the same time it's going there to be integrated with memory, it's also going with the amygdala, which is going to tag that memory with a hashtag of emotional valence. Is it a good smell or is it a bad smell, right? It's like the Wizard of Oz. Are you a good witch or a bad witch? And so you can see how this stuff kind of comes together, right?
I was gonna say, in terms of like disgust, is that more of a smell thing or a taste thing, or is it a combination of both, or?
It's probably a combination of both, but definitely involved in taste. You can look at the cortical areas that end up being involved in processing some taste perception. They're called gustatory cortex. Gust, as in disgust.
What about when people hallucinate smells?
Good question. Hallucinations.
So like, do these areas kind of still come into play, or is it something completely different?
Yeah, I think so. It's probably going to be more in these cortical areas
Mhm.
that it gets sent to. Let me see. I might have some maps of that here. Yeah. So these are the same areas we looked at earlier, right? You have your orbital frontal cortex, the piriform amygdala, but we can see that it's also going to go from these areas, right? It does go to the thalamus, just doesn't go there first. But a big spot it's going to go to is the orbital frontal cortex. And we talked about this entorhinal being the anatomical pipeline into the hippocampus, right, for memory formation. But look at where the rest of this goes. The big thing is the thalamus, and the thalamus is going to crosstalk. It's going to go back and forth with the orbital frontal cortex. That's what's going to allow it to back and forth, because we're figuring out what is it, right? We're testing out, so this smell is this, what does it mean, right? Not just is it good. First we got to tag it into memory or retrieve it from memory, right? So I'm smelling something. Is this something I've smelled before? So, I can't hear or see this guy dancing behind me in the bar, right? I can smell something on him. Is this a smell I know from the past in memory before, or is it a new one? I have to know that right off the bat, right? And then I got to figure out at the same time, is it a good smell or a bad smell? So it's a good smell, like, oh, I haven't seen my boyfriend in a while, he's really great, right? Like, it's a very comforting smell to have, right? Or is it a bad one? So you got to figure that stuff out, very early primitive information, right? Emotion and memory. At the same time, then we're coming back and forth, back and forth, back and forth, and more higher thought processing, right? Okay, so it's the same smell that I've known before that he does wear, but is he deployed somewhere? Is he even in this state right now? So it could just be somebody who's wearing the same thing. Like right now we've done the identification of the basic important elements, but now we go back and forth with our cortical processing of what it is and specifying the details, right? What you might think of adding context. Very important things, right? Because we're not animals. We're not robots. What makes us special? Well, we have the ability to understand context. We can't program robots in artificial intelligence yet because we haven't figured out a way to make machines do what humans can do to detect context. Right? Any of you guys ever seen the Johnny Depp movie called Donnie Brasco?
Where he plays like a
Mobster.
undercover?
Yeah.
Yeah.
Anybody? You guys know what I'm talking about? He's playing an undercover FBI agent for the mafia in New York in like the 70s. And so his character is very much this like Italian mafioso with a New York accent. And part of the way these guys talk is they use the F word in everything. No, actually it wasn't the F word. They say forget about it. And there's this famous scene, YouTube it or something, where one of the other FBI agents is taking him out for a drink to debrief him. And he's like, what is this thing forget about? You guys say this about everything. And Johnny Depp goes through this, he teaches him basically. He's like, yeah, forget about it. It means whatever you want it to mean. It can mean go screw yourself. It can mean, oh, don't worry about it. It can mean 100 different things. But the context, so if you say forget about it to one of those guys, and it's to an artificial intelligence algorithm, machine learning, or a robot's not going to know what version you're talking about, right? They're not going to know if you're joking around. They're not going to know if you really mean go screw yourself. They're not going to know what you mean. Humans are special. We can process context, and that's what's happening here. Contextual processing. We can assign what it means in the context of what else is happening, right? So that we don't act like idiots. Be like, oh, yeah, you said this. So, no, come on, you know what I meant. It was context, yeah, You guys follow what I mean? Okay. Okay, so, this is just showing the differences evolutionarily between, say, dogs and humans, right? So here's the cortex devoted to a rat's smell. This is our olfactory bulb in humans. Not just, are they bigger, but look at the proportional difference. But that's kind of old hat. This is just sort of illustrative. And you can use, right? So this is really funny. So this is an actual experiment that people did. And you can drag what, oh my goodness, if this was Juno, she would lose her little doggy mind. You can drag a pheasant through the field. Juno's a greyhound, so she's like bred to sight hound birds. She loses her little mind when we go to the park and chase birds. And drag it through the field, right? You see this yellow line, and the dog will go back and forth, and it will use sense of smell to localize. Humans can actually do the same thing, but not for pheasants. You know what they did here? They used chocolate.
Mm.
Yeah.
Yeah, does it matter that, like, the pathway in the dog was take, seems like a lot wider than the pathway for humans?
Yeah, it's a little wider. I'm not sure that it matters. I think one of the two things I remember being communicated about this data was, one, the fidelity to the actual track, right? So it's not a random hodgepodge looking around and then, oh, even a blind squirrel can find a nut every now and then, right? It's not just like, oh, you got lucky, oh, you find this thing over here because you looked everywhere. It's a systematic, they are following the track. But the other element is that not only are they following the track, but the way they follow the track is not to follow the track. It's to crisscross. And so you're constantly going back and forth, recalibrating. You go left, you're zigzagging essentially.
Yeah, that's what I was meaning, sorry.
Yeah.
Because the crisscross seems to go a lot farther away from the actual track than the criss is. Like, does that mean anything?
I don't recall off the top of my head on that particular element. But yeah, pretty cool stuff. Let me see if I can find some more of these pictures that I was looking to show you guys. This is that vomeronasal organ in the rodents. Okay, so it's very similar. They're going to have, instead of G olfactory proteins, they're going to have VR1s, vomeronasal receptor 1, or vomeronasal receptor number 2. Different protein structures that are going to respond to those odors. Okay. This is an actual micrograph of the cilia in our olfactory receptor neurons. Okay. And again, we looked at that Bowman's gland. I did promise that we would come back to that. Okay. So secreting that ooey gooey mucus. Okay. So you can think Bowman next time you sneeze on somebody's pooping. And what we can see here too is stem cells, right? Neurogenesis. See little baby stem cells beginning to differentiate. And then we can see them start to climb up and integrate into our sustentacular support cells, right? If they haven't really integrated, they haven't even crossed out into the mucus. Here, the next one over, they grow a little further, they start to get out there. Now they can start to get their cilia out there too. So it's showing that temporal progression too. So we have a lot of stuff, yeah.
Is that mitochondria going from the sustentacular cells into the ORN?
No, not that I'm aware of. I don't think that's mitochondria.
Okay, what are those?
You mean those little red dots?
Yeah, what are those?
To be honest, I'm not sure off the top of my head.
Okay.
It's a less important thing to focus on for this diagram.
Oh.
It's not going to matter so much.
Okay.
Now, what you'll notice too is that this diagram is upside down from the way we've typically been framing it. But that's okay, right? Because we have a vestibular system that allows us to roll upside down and still know where we're at. Right? It's true. I was talking to a guy yesterday who was going to do some aerobatic flying. He's going to take me up. We're going to do some loops and rolls and all sorts of crazy stuff. And you've got to have your vestibular system. So, you guys, I'll show you some. So when you fly, you have, let's see here. You guys want to see this?
Yeah.
Okay. Let's see. This way I can draw, but the lights can stay there. Okay, so really quick, when you fly, you have a gauge. It's called an attitude indicator. And it has wings on it, essentially. And this will be essentially the ground. And this is sky, right? And you're going to have little marks on it. And you're going to have your little airplane wings. And you're going to have a little nose of your airplane, right? Remember we talked about pitch? So this will tell you pitch. So if you're pitched up, it'll actually show that it's higher than the ground level. It'll show you're pitched this angle. So it tells you, as you're flying, which way you're going. Because maybe you're in the clouds and you can't see where you're at. So you need to use your dials to learn, right? So you don't crash. Same thing, if you're pitched down, right? You're gonna have your little airplane's nose and wings down here, right? Because this is your ground level here. You guys understand? So the point is to your question about the cribriform plate and all that structure look the other way. When you fly upside down, right? That's what your dial looks like the other way. Such that we'll make it easier. Sky is always blue, right? But when you're upside down doing aerobatics, your dial looks like this. And it's how your window looks too. Because you're upside down, right? And the point that I was making from it was that just like when this happens, you've got to not panic and just know, okay, we have a vestibular system. I know that I can hang this way and still make sense of where I'm at. That's all. So that's just a little stupid tangent, essentially. But by the same way that we can do that, Corey, can you hit me on the slide? When this flips around, it's the same way that we can not get, hopefully, not confused when we see that we need to rechange our orientation. So here it's just taking the plane and going upside down. And not going, oh my God, this is a whole new thing. Oh, wait, I thought this stuff worked this way, and oh gosh, I'm so confused now. No. Right? Okay, so let's see if we can see some other pictures. Same things we've talked about. I'm relying on you guys to slow me down if there's something you want me to stop for because we've covered this stuff already. Let's see here.
I do have a question.
Yeah, hit me.
Okay, so when the odorant comes in, does it enter the mucus layer before hitting the receptor, or are the receptors sticking out? Like, are they sticking out past the mucus layer to get bind?
The cilia is in the mucus.
So the odorant has to get
has to-
inside the mucus
yeah.
and then get to the, okay. Alright, thanks.
Let me see if I move here. This is that process again, transduction. You have a molecule G protein coupled reaction, right? ATP, cyclic AMP, opens up these channels. Whoop, looks like calcium and sodium. Those come in. And we got the chloride action there too. So just another way to look at it, right? It's all cartoons. It's easy.
If I were to touch my face, where would the cilia be? Like, is it like, I'm just trying to like think like anatomically, like, is it like
Never been asked that
in
before.
past?
It's a good question.
Or is it like up here
Well, let's take a look.
in like the sinus area?
Maybe, yeah, it would be here. So,
So,
right.
yeah, but like,
So this is teeth, nose, right? So it depends on how big of a schnoz you have. Right, if you're Scottie Pippen, right? You guys know who Scottie Pippen is?
Yeah.
Okay. I didn't know if that was too old of a reference. But I grew up in Chicago as a big Bulls fan, so I knew Scottie, and not personally, but he had a commercial where he had something where he had make a joke of himself about having a big schnoz. And but let's imagine it depends on how big your nose is, but this is probably about eye level, right? Ish, right? You figure where's the nose kind of, so this is where kind of your bone kind of comes back. This is about here.
So it's
So
kinda like behind your eyes or like within like.
It's like in the center of your eye.
yeah,
Yeah.
right? So it's at the base. It's at the base of your orbital frontal cortex, right? So it's not gonna. And your forehead is basically the base, right? Because your brain isn't gonna come beneath your forehead, essentially, right?
Yeah.
You get your eyes and your goop and whatnot.
Yeah.
So
Yeah.
it's about here-ish, maybe.
It's like temporal.
Ish.
So if you had like trauma to that area, could you damage your olfactory
Yeah.
system?
So that's a really good point. So this happens. So remember, I said you can get anosmia from chemicals. You know, if you get exposed to a bunch of ammonia and the toxicity brutalizes those cells or something. There's times of year when people have been in bad car accidents or bad, just messed up stuff happens in life. You get injured, like some crazy thing, and you can actually have so much force that you either can break the cribriform plate or it cuts the axons that come through it by either rotational shearing forces of an injury, like a really heavy whiplash type of thing, or concussion, or it actually breaks the bone itself if it's a hard injury, and then you can't smell. Very different reason for why you can lose that sense. Just like there's different reasons you can lose your sense of vision, right? You can have a cortical blindness, which means that there's damage to the visual cortex. Your eyes work just fine, but your cortex can't process it. And then there's, of course, you can just have your eyeballs plucked out. You can damage your retinal ganglion cells. There's all sorts of ways that you can have damage that are going to affect the visual system differently. Same thing. There's different reasons you could lose your sense of hearing, right? So yeah, really good point about that. Let me see if I can find some other cool pictures that illustrate these things. We've talked about this process. And then, of course, we've got a sodium-calcium exchanger that'll help to reset that afterwards. That's a little extra bonus for you.
And that one's just 1-to-1.
It looks to be the case. It looks to be the case. So, this is way more complex than you guys need to know, by the way. So that's why I'm skipping through all these complex slides for now. So, remember I said I'm just looking for the right pictures to help dial things in. A lot of this is showing that glomeruli again, right? So you can image these glomeruli. This is a Nissl stain, right? So you have that glomeruli here, okay? Let's see here. I'm looking for some other, so there's mitral cells, and I'm gonna show you a cartoon that shows them much better in a moment or two. But this is an image of these glomeruli, getting those projections. Okay. So just, enjoy the art for now. We work through here. God bless you. So this is a nicer image. Okay, so mostly everything is the same. Okay, you don't need to flip the plane upside down. We're still looking at it the normal way. Cribriform plate, glomeruli, but this shows some other cells in the mix, just like we have horizontal and amacrine cells in vision, right? Right? We've got some mitral cells and tufted cells that help to modulate. So they're the ones that's going to send their secondary, these are the secondary olfactory neurons, right? That we started off generically calling secondary olfactory receptor neurons. Now we're giving some extra specificity and names. Mitral cells, tufted cells. They're what's going to send their projections out together with some granule cells to the lateral olfactory tract. Okay? So let's back it up again here. Right? Here's our Nissl stain, and here we have our glomeruli, our mitral cell layer, our granule cell layer. Okay? Just like we saw here. You have glomeruli, a layer of mitral cells, and a layer of granule cells. Okay? And this is stained for just one glomeruli, and you can see all the axons of the ORNs projecting into it. Pretty cool, right? Yeah. And remember I said it's kind of like the lights on a marquee? Okay. So you can imagine this is just one, but you can imagine lots of them lit up, and we'll see that. So that's what we're looking at here. This is a way to image some of those glomeruli. And these are different smells: ethyl acetate, eugenol, isoamyl acetate, acidophenone with a higher concentration. Okay? We can see that they're activating different glomeruli, different patterns, right? These are like little light bulbs on the screen, analogy-wise. So that's pretty cool. Again, this is another way to image those glomeruli. That's what these blobs are. Okay? And notice that both of them are responding to the same stimuli. Okay? And that the bigger the concentration, right? So this is our concentration, little bitty baby bit, 0.00001 to 100% full-blown smelling. It's really drawn bigger activations, but it's still these two. And as you get to these higher ones, you've recruited a couple extras to the party.
So because the radiuses we're breathing in, right, through our nose, but we're not consciously paying attention to what we're smelling. Is that possible? Or
Happens every day, right? Absolutely. I mean, that happens with vision and every sensory system. I mean, are you really paying attention to the signals your butt is giving you right now from how hard the bottom of the chair is? I mean, maybe, but yeah, right? But you weren't. That's exactly the point. Were you getting those signals? Yeah. But were you paying attention to them? No. So we're always detecting things, right? Like, you guys know, obviously, I don't think we're robots, at least I'm pretty sure that I'm not. And, but, right, you know, I obviously have a profound appreciation for our humanness and our humanity, so I don't like to consider us like robots and analogies, but if you think of us as these machines, right, as we're nothing more than machines with systems optimized to detect things in the world, and not just things in the world. Remember when we talked about cell signaling, we talked about not just detecting things, but detecting changes in things, right? So changes in how hard that seat is on your butt when somebody brings it up, because otherwise you weren't paying attention to it. It didn't change, right? It wasn't any softer before that, but it was a change in your recognition, not in the change of the physical stimuli. And so we have all these sensory systems, vision, audition, right? We're always taking visual things in. Are we always paying attention to them? No. Are we always smelling things? Yeah. Are we paying attention? Most of the time not, right? Doesn't mean they're not getting into our brains though and being encoded as memories. And we'll talk about the unconscious ways that we still put that information into our nervous system. And in fact, if you don't believe me, then you should bet against every advertising agency in America. Because the entire billion-dollar industry of marketing, advertising is built upon the facet that your behavior will change and you will remember things even if you didn't actually consciously notice it. Right? And if it didn't work, they wouldn't be investing billions. So we do notice those things, I guess is the point. And so let's see if I have, these are mostly the same pictures, the same diagram we've looked at before. We've seen this diagram before as well. These are different ways of showing that. So here you're smelling these flowers. We've got olfactory receptor neurons, cribriform plate, glomeruli, mitral cells projecting through granule cells out. So same way to look at that, a little bit differently drawn. Here's a nice little anatomical picture.
So do the odors, I see right there, they bind to these sites, do they kind of act like neurotransmitters a little bit?
Exactly. Yeah, so they're just ligands, essentially. And again, what are they going to do? They're going to open up those receptors and allow that signal transduction process to happen. Not by opening these necessarily, but by this would be GO. It'd be inducing that cascade of effects that we talked about. Again, these are similar anatomical drawings if you like getting into the nitty-gritty. If you like more teeth, you can see them down here. You know, anybody wants to know why you get your wisdom teeth out, right? Those are going to be those suckers back there. Yeah, sorry. Yeah.
After the odors bind, do they what happens to them? I mean, neurotransmitters either get reuptake or
Yeah.
degradation. What can they bind again to another receptor? Do they just float away?
They can, all those same things can happen. They can be degraded by enzymes, they can be essentially unbound by other things, competitive binding. The cell is definitely gonna reset at some point. Let's see here, again, different illustrations of the same points. We've seen this micrograph. This is that process again. Just shows the steps of that binding. Some people like different ways to look at it. This is a really cool illustration of that vomeral nasal organ, showing that you can stain for different receptors in different parts of the olfactory epithelium. Actually, not the let me correct it. It's not the vomeral nasal organ. It's the olfactory epithelium of the road. And you can see that you can stain for these different types. Every color represents a different gene for a different type of receptor. And not only can you see them illustrated across here, but you can see them projecting essentially differentially to the olfactory bulbs and the glomeruli. And this is that marquee element. So you see these glomeruli lighting up in different ways, essentially representing the convergence from those different types of neurons, which I think is really cool. Yeah.
So the organs, there is a systematic organization. Oh yeah, okay, I was thinking that. Because everything looks like your receptors in your eyes, your ears, there's always a systematic organization. It looked pretty random.
Yeah.
At
I mean,
first, but
it's
that
really
was-
easy for us to think of
Yeah.
like these evolutionary things are like, oh, it's just a bunch of random hodgepodge, like
Yeah.
this sort of Darwinian randomness, right?
Yeah.
But like every time I just get more amazed and amazed and amazed. Every time I learn more about these scientific depths, there's more systematic organization than there is this random hodgepodge. And it's elegant. It's really beautiful.
But that slide kind of illustrated because your other slides, I was trying to figure out if there is or how that's shown.
Yeah, well, what would you have done if I started with this slide? Right? Like, this would have been overwhelming, I think.
Or no, the previous one, the one where it showed that the staining. I was like, oh, okay, that's pretty.
Yeah.
Because that shows it, how it stains it and how it gets to the vortex and how it's mapped on there.
Yeah, it's really cool, right?
Yeah.
So, yeah, and then, you know, you can get computational models, same information, you just get the anatomy out of the picture. You just see how it projects and converges, so that's pretty cool. You can bring our good friend Golgi back into the mix, right? 5% of the cells, the whole body. See those going up to the olfactory receptor bulbs. This is pretty cool, you know, color diagram. Here we're upside down, so you gotta flip your airplane, right? Anyways, you can see how people model this stuff. And here's the real deal, right? There's our olfactory bulbs, come in here, medial temporal lobes.
So we have two?
Yeah, left and right.
I was hoping I thought we had one.
Yeah, I thought there was just like, okay.
I'm glad that we found this, right? So do you guys want me to put these extra picture slides up there?
Yes, please.
Okay, so I'll put these up there, but I'm not gonna have time to trim. I'll put both files up, but I'm not gonna have time to trim them to cut out like the taste and chemoreception stuff. So I've had people before complain that like, oh, you put so much slides up there. I'm like, yeah, but you only gotta know the ones that we talked about. Like, so don't worry about having to know the chemo, the taste stuff or the hunger stuff, because we haven't built that into this class. So just use this for the illustrative purposes that it's here for the types and the ways that we talked about the material, and don't let it overwhelm you in that regard, because that's only here because you guys asked to have that extra supplement added in. So use that as a good thing, not as an overwhelming thing. I think next Monday, I'm definitely not going to be here. I'll be at a Society for Neuroscience conference in DC. So we will either cancel class or I might send a video of a lecture that had been delivered prior on a topic that was otherwise scheduled. So keep an eye out for the email. I'm not sure which one I'm going to do yet, but either way, if I send a full-length video of a full lecture presentation, then then that'll be information expected to be responsible for, and you can bring your questions about it. But I'm just gonna take a closer look and see if I can find something good, and if not, then we'll have the day off. If that sounds okay for you guys? And then,
If we have the day off, could you just send us the video of the lecture anyway, so we could review it at home?
well, maybe, I have to go figure out what topic I want it to be on exactly,
Okay.
and if I have a good video of that topic, and if I don't decide that, then I won't send it.
Okay.
But either way, we're not coming.
You can come here and watch it.
Well,
I mean, you guys should seriously get together and practice and talk about this stuff. Practice drawing it, like, we're gonna have more class participation stuff like we did today, but more and more. You should be able to start drawing these things and integrating them. And I'm gonna ask you that stuff. And if you guys don't have questions before we start from what you learned or what you studied beforehand, I'll consider my job done. I'll go home. I got other stuff to do in the morning. Like that was, remember the core part of the sort of philosophy of the course is that I'm expecting that the material's prepared, not perfected, but prepared before we get here. And my job would be to help you guys clarify and understand what you're prepared with. But if you guys don't need any help with that, what do you need me for? It's gonna be my attitude. And so have that stuff come, and it'll help you guys integrate that stuff. And remember that there's a whole 5th grade in the class on participation. So some of you guys are really good at that. Some of you guys haven't participated much, and I want to make sure that you guys can benefit from that opportunity to score points. So don't miss those opportunities, because they're going to be otherwise associated with bad grades. Because hopefully you guys see that the more that you guys integrate and participate, I'm able to help clarify your understandings, so you actually get it better and do better. So trying to help you guys out with all that stuff. Put this stuff up here. I appreciate your patience, and we'll see you guys next Wednesday. Yes.
I noticed there's a video about English yesterday, and when I went to look at it.
Oh yeah, there's no bar.
Yeah, I was like, there's no bar.
Yeah. Sorry.
Because I was gonna use it for some of the questions, and I was like, oh. And I was like, no bar.
I try, but sometimes it's like fails.
That's okay.
Question please. If I send you a link to a book, could you upload copies of the audio recordings? Yes. One of the other students just asked, I found a video I uploaded
No,
yesterday.
I just had so much
Doesn't
more.
have the audio.